So can we start, Jeff?
Yes.
Okay, so I think everyone is here. I want to welcome you back, and I want to thank you, Jeff, for having accepted our invitation. It's an honor for us having you here this afternoon for us in Brazil, morning for you in the US. It's a real pleasure having you talking about Brucella and all the evolution of the genome. As I said to you, we are going to use Brucella as a model for our course. People here are from all parts of Brazil and South America countries, and also from Cuba. And thank you so much. They are all yours.
All right. Yes. Thank you so much for the invitation. And it is my great pleasure to be talking to you all. So perhaps we'll leave it to the end for questions or put questions within the chat. Yeah, this is, I don't know how you typically do that, but that may
It's the same. It's typically at the end of the presentation. They save the questions for the end.
Okay, sounds good, and we can go back to particular slides if need be. Yeah, so I'm going to do a brief introduction to genomics, not in incredible depth, but I'll show you kind of some of the history of how we understood and understand how Brucella has evolved and the genetic techniques that were used in the past, and then go to present, how we're using kind of state-of-the-art genomic techniques to assess Brucella genomes. And then we can see the application of genomics to understanding both epidemiology at relatively short timescales, and then really understand or try to understand how Brucella has evolved over thousands, perhaps tens of thousands, perhaps hundreds of thousands of years. And so there's a bit of uncertainty about the timing, and we'll discuss some of that. And that may be kind of food for additional conversations later. And so I love this view of the globe. And so we can really think of Brucella. I think Janira is live.
Não, porque o meu não dá pra tirar, porque olha, fica assim, ó.
And I love this global view of Brucella or the globe, and to really think of Brucella as a global disease. And indeed, it's amongst the most successful organisms out there, and I'll show you that in a second. And so this story kind of begins and ends in the dirt. And so remind me if I forget to kind of close the loop on that. But indeed, all of Brucella's closest relatives are in the soil. And at least in modern terms, the story of Brucella begins on the island of Malta in the 1880s, where a British attachment was stationed there, and a bunch of the soldiers that were stationed there on the island of Malta, and we can see it here kind of below Italy in the middle of the Mediterranean. A bunch of soldiers there became ill, and Sir David Bruce was the lead surgeon there and was able to identify that a particular bacterium had caused this disease. And so there's actually a lot of debate over who's truly responsible for kind of this discovery. David Bruce gets most of the credit, but in actuality, it seems like it was a team effort, and several people were involved on both the animal health side and the human health side in identifying that. And so we go back, and so indeed it was later connected to infected goats. And so this was the first kind of evidence of Brucella infection that was transmitted from animals to humans. And so we go to present day and really using genomics to try to understand the spread of different pathogens. And I think SARS-CoV-2, maybe I should have a slide for that as well, that is a great recent example where we can actually use genomics to understand the spread of different pathogens. And so something like SARS-CoV-2, we can use genomics to look at different variants and how they spread globally. And a great example is the Delta variant. I think everyone is likely familiar with that variant. So that variant is just one of many lineages of SARS-CoV-2. And so we can use that same approach. And in my case, we typically work on bacterial pathogens. And so we can use genetics and genomics to really look at how pathogens spread globally. And so one example of this has been used in the past is Helicobacter pylori. And so it has spread globally with the movement and migration of humans. And so in our guts, a bacteria has been transported alongside us and has basically gone global. And in another case, we have, again, with the spread of humans, we have Mycobacterium tuberculosis. And so this is typically causes pneumonia. And so we have spread TB globally. And a great final example here is that is the spread of bubonic plague or Yersinia pestis along with our black rats alongside our shipping. And so we have 3 examples of different bacteria that have been spread globally, 2 by humans and 1 indirectly by humans through commerce. And in each of these cases, we can use genomics to track the spread of the particular pathogen. And so, stories about genomics deal with 2 major themes. And so we're really interested in time. And so using phylogenetics to understand time. And so time is usually, as we'll see in subsequent images, time is usually represented on the x-axis in the horizontal. But we also are interested in how pathogens have spread geographically, so across space or across geographical areas. And so much of my work tries to tie time and space together to really understand how pathogens have spread. But before we get to that, and so that's much more recent, we're trying to understand the past. And to understand the past, we really need to dig into the genomes. But if we look at Brucella out there, if we look in various livestock and domesticated animals, we see that Brucella is extremely widespread. And I'll show you 2 case studies, one in cattle and the other in goats, about how successful it's been. But it's been spread globally in our domesticated animals. It's widespread globally in various wildlife that are out there. And it's also in our oceans. And so amazingly successful bacteria that is in a full range of habitats from the Arctic to the tropics, from the oceans to the, nearly the highest peaks. And so a very widespread and successful group of organisms. And so one common theme that you'll see here is that this is a bacteria that has been able to successfully infect a broad range of vertebrate hosts. And so in the past, we used to think that they would only be warm-blooded hosts. But we now see that various fish species and amphibians are infected as well. And so the more we look, the more we're finding Brucella in a broad range of different hosts. But it does seem that that this is an organism that has adapted for vertebrate hosts and has many adaptations specific to living in vertebrate hosts, and has lost many of the adaptations that its soil relatives have. And we'll see that in a second. So indeed, as I just mentioned, all of the close relatives that we know for Brucella are in the soil. And a great example of this are the Ochrobacter species. And so Ochrobacter is a genus of bacteria that is in soils worldwide and are the closest relatives we know of to Brucella. But something allowed Ochrobacter, or the close relative of Brucella, to emerge from the soil and infect a broad range of different hosts. And so back to kind of the main focus of this is how do we take the actual microbe itself and really understand how it's evolved and how it has potentially spread. And so we do this by culturing the organism. At least we had to do that in the past to do that. So we take Brucella grows pretty easily on standard sheep's blood media, and then we extract its DNA, and then we look at its DNA and try to understand the relationships of different strains that we may have. And so early on, so this is from 1985, and this was unfortunately very, at least unfortunately in my mind, very influential article where Verger came out and said in 1985 in the International Journal of Systematic Bacteriology that Brucella was a single species. And so it was a monospecific genus, and everything was a single Brucella melitensis. And so this was very influential and has fallen, I guess, into disfavor since then with more advanced genetic and genomic techniques. But this was kind of the beginning. And one of the major challenges, and it exists today, is that Brucella, at least compared to lots of other bacteria, isn't very diverse. And so it's relatively monomorphic. And so genetically is very simple. And so lots of the strains look, at least on first pass, very closely related to each other. And over time, we've gotten better and better tools to be able to look for the variation, the genetic variation that exists, and really identify different groups. But part of this really goes back to kind of differences of opinion in bacterial taxonomy and how you define a species of bacteria. And not everyone agrees on that simple idea, or relatively simple idea. And we're not going to kind of solve that right now, but I will detail kind of some of the evidence that we've generated to look at that variation. And so this is one of the initial studies trying to understand the genome of Brucella. And I think the next talk, or a talk later today, is going to be on Brucella chromosomes. So I won't go into too much detail, except to say early on, several French groups used restriction enzymes to map out the different chromosomes of Brucella. So their first study in 1991 actually only identified one chromosome. And then a couple of years later, they realized that it was two chromosomes, a large chromosome 1 on the left of 2.2 megabases, and a smaller chromosome on the right, roughly 1.15 megabases. And so in total, the genome of Brucella is roughly 3.3 megabases, or 3.3 million bases. So early work, seems pretty crude right now, but they were able to, basically map out the chromosomes and do really great stuff with relatively, crude technologies, at least compared to what we have now, but really groundbreaking at the time. And so what are we trying to do here? And so I'm just trying to show this more simply because I think it's difficult to imagine how you compare two chromosomes when they're circular. So I've just simply snipped those chromosomes at one point and laid them out down flat. And so how do we go about doing this, comparing two or more strains against each other to really understand how related they are? And so this is a simple way. And so we may want to do, and I think this is relatively intuitive, is we may want to just line up another strain end to end compared to the first strain. So we have two strains compared against each other in two different chromosomes. And we just simply would go base by base and compare them. At least theoretically, we could do that, right? And so we have a comparison. But it becomes really complicated really quickly because bacteria don't keep the order of their genes exactly right, especially from strain to strain. So if we tried to do this comparison, we really couldn't do it because of the shuffling and recombination that occurs even in an organism like Brucella. So this is just a simple example. We can see here that this one gene on the left-hand side, ACCD, has moved in the top two strains. It has moved to the far right in the third strain, and then in the fourth strain, it's moved back. And so this was caused by an inversion. And so this is just one example of how the genome can get reshuffled and how we can't use just a simple straight up comparison of two chromosomes against each other to understand the relationship, because there's this shuffling of genes and shuffling and of the genetic material around different chromosomes. And so this is even a better example. So this is more complex, and we're comparing, in this case, Yersinia pestis, but you get the idea that there's lots of genes moving all around. And so this is a comparison of 9 different Yersinia pestis genomes, and we can see great reshuffling of the different genes. Nearly all the same genes are present in all of these isolates, but they're in different places. And so it becomes very difficult to try to assign a gene by gene comparison. So the first attempts at doing this, this was well in advance of our relatively advanced genomic sequencing techniques that we have now. We went by gene. And so the first work in most organisms, or all organisms genetically, was to look at a single gene. And so in humans and other vertebrates, you can imagine various mitochondrial genes and comparing mitochondrial genes against each other. And so Tom Ficht and his collaborators in 1996 used a single gene locus, the OMP gene, which is an outer membrane protein gene, to compare different Brucella. So one of the first kind of phylogenetic comparisons, and it was using a single gene and sequencing of that gene and comparing. And so we get various relationships here. And so we get that Brucella canis and suis are related to each other. We get abortus and melitensis kind of mixed together. And then we get more distantly related Brucella neotome and ovis. And so a great first attempt at trying to understand the relationships of different Brucella, but it was only one gene out of thousands of genes, right? And so out of thousands of genes. And so additional researchers kind of used other strategies. And so in 1997, only a year or two later, using restriction enzymes and DNA hybridization, this phylogeny was developed. And amazingly, despite again, relatively crude techniques compared to now. This phylogeny and the relationships of different Brucella is largely true now. Same rough branching structure then as now, and we have entire genomes now, all 3.3 million bases to compare, and we still get the kind of same pattern. So at least it's pretty amazing to me that using agarose gels and a binding of DNA to between different strains, you can kind of repeat the fine scale structure that we even know today for Brucella. And this is just an additional kind of similar approach. And this is a bunch of related organisms to Brucella, and we can see here Brucella melitensis. Hopefully you can see my cursor. Brucella melitensis is here in the kind of bottom center, and then we see the closest relative, at least in this phylogeny, is Ochrobactrum anthropi. So Brucella is just one of many different soil bacteria that are out there, and so all of these others are soil bacteria, and its closest relative is Ochrobactrum, and it's just one of a whole large group of soil bacteria. And this is more recent, but again, using gel-based relationships. And so this again identifies Ochrobactrum as the nearest group, and then we can get some differentiation of different Brucella. Again, really similar to actually past results, using a different approach, but we can identify different Brucella species, and we can identify some of the relationships amongst different species. And so that's the kind of the past work, or that's the past work using genetics to try to understand the relationships. And now we have, I would say, more advanced or more sophisticated approaches at looking at the relationships between different strains or different species of Brucella. And there's been two primary ways that we've done that. The first of these is using variable number tandem repeats, or also known as VNTRs. In the vertebrate world, they're called microsatellites. But the basic idea is that you have different numbers of repeats. And so you see in this example, the first strain has GATA repeated 4 times. The second strain has it repeated 3 times. The third strain has it twice, and the last strain has it repeated once. And so these repeats actually happen fairly frequently, or repeat differences happen fairly frequently. And so we compare different strains based on not just one locus or one region of the genome, but we look at many parts of the genome that have repeats, different repeats. They won't have this exact kind of range of different repeats, but we look across the genome to see which strain is most closely related. And so at a particular locus or at a particular region of the genome, 2 related strains will share the same number of repeats pretty commonly. And so say the most commonly used analysis uses 16 loci, so 16 regions of the genome, each with a different kind of arrangement of repeats. And some of those strains are going to be identical across all 16 loci that we have, suggesting that they're very closely related to each other. And the more repeats that are different, or the more alleles that are different, the more diverged 2 strains are. So that's VNTRs, and we use it in this big analysis called MALVA, multi-locus variable number tandem repeat analysis. So we use an acronym because that's a mouthful, but we're basically using a whole bunch of VNTRs to look at the relationships. And so this is the most common approach, partially because it was developed first, and partially because you don't need whole genomes. It's done relatively easily with a capillary sequencer, or you can even do it on agarose gels, and you can see the length of the amplicon, and the length of the amplicon indicates typically how many repeats that it has. And so you can determine, you can use that as a marker. More recently, and much of this talk is going to kind of focus on this, although we'll occasionally go back to VNTRs, we can use SNPs. And so SNPs are single nucleotide polymorphisms, and they're simply point mutations. And so we can see here, at least at one locus, so one position in the genome, strains 1 and 2 share a nucleotide C, and strains 3 and 4 share the nucleotide T, right? And so if we just used this evidence from this locus, we would think that strains 1 and 2 are closely related to each other, and 3 and 4 are closely related to each other, and that they're different from each other. But we tried to never base that on a single locus. We look across the genome for all of the SNP differences. And so if strain 2 share, the majority of the SNP differences, that would support the likelihood that they're closely related to each other and less related to strains 3 and 4. And so we use that to develop, particularly we use SNPs, although we can use VNTRs as well. So just looking at how strains are related to each other. And so this isn't actually, this is actually a mammal phylogeny, but the basic idea is the same across kind of phylogenetics, is that as we get closer to the branch tips, we get closer in time to the present, and that these are recently derived relationships. As we go further back in the tree, further back in time, and so we typically assume that as mutations accumulate, and so mutations accumulate in general over time, we can actually estimate when particular differences emerged. And so in this tree, these are deeper evolutionary relationships that happened longer ago. And these kind of more towards the branch tips here happened much more recently. So again, to emphasize this, we typically use this x-axis as a measure of time based on the number of mutations that have accumulated at roughly the same rate, at roughly a constant rate. And so we can perhaps kind of talk about that assumption later, but in general, it holds true with a few exceptions in that mutations accumulate over time, at roughly a constant rate with kind of a few exceptions. And then we can understand when the various divergence has happened over time. And so just to kind of remind you of what we're talking about here. And so we're talking about Brucella, and Brucella is a clonal organism. And so what makes that really nice for Brucella is that when we see a mutation, all of the ancestors after that mutation, and so all of the offspring from that parent are going to have that mutation. So we see a mutation that happens here, and we see actually, in this graphical representation, we see a change of phenotype from blue to green. And then so all offspring after that will have that green phenotype until there's an additional mutation. And so what we know is that we can basically use the genetic changes really identify who's related to whom. And so we have pretty good certainty. What is difficult in other bacteria, and it's true in viruses as well, is that we have recombination. And so we have the movement of particular genes from one lineage to another. And so that kind of obscures the relationships. And so that makes it very difficult if you are using a particular gene to look at the relationships, but that gene hops from one lineage to another or one species to the other, you would get confused about who's related to whom, because you would think that two unrelated organisms are related to each other just simply because they share the same gene, but that gene was horizontally transferred or moved from one group to another. In this case, we don't have to worry about any of that. That almost never happens in Brucella. It's clonally propagating, so the mutations that we see are fixed until another mutation occurs. But then we can even use that to look at the relationships and understand those relationships. And so two things that we think about, or the main thing that we think about, is that mutation rate is happening over time, but mutations and how they occur don't happen at the same rates. And so actually these VNTRs that we were talking about have a pretty broad range of mutation rates. But the SNPs actually evolve more slowly. And so this is one base mutation per measure of time. And so we have VNTRs are accumulating much more rapidly in general than SNPs. But the benefit of SNPs is that we oftentimes have hundreds, if not thousands of them. And so even though they evolve more slowly, we in general have a lot more of them. And so we have a lot of power to look at the relationships of different isolates or strains using SNPs. But VNTRs kind of have their role as well. One challenge, though, is that VNTRs evolve so quickly that sometimes they evolve, too fast. And so we can't use them for really changes that happened long ago, because it's equally likely that those changes have reversed or reverted to what they were previously. And then we are confused because those markers are evolved and then evolved back. And so we can't see the relationships between different isolates if that marker is not consistent. So this was a tree using VNTRs in 2006, and this was a really influential paper. And basically much of modern epidemiology for Brucella uses this same approach. And what's great here is that, and we can see, if you roughly remember back to some of the early DNA hybridization work, it's roughly the same tree. And so we have groupings using VNTRs, groupings of Brucella abortus here, and I'll show the significance of that in a second. Brucella ovis here, Brucella melitensis here. And so we have pretty good groupings of different Brucella that are out there using these relatively rapidly evolving DNA markers called VNTRs. I just thought I'd show this quickly. This is a bit off topic, but there's other ways that we can do this. We can use, rather than VNTRs and rather than using SNPs from whole genomes, we can use single genes, or we can use several genes. In this case, it's 7 genes that have been used. And so this is multi-locus sequence typing. So basically, the sequencing of 7 genes from across the genome, and we roughly get those same relationships. So again, to kind of drive the point home is, regardless of the genetic approach that we're using, we're getting roughly the same relationships of the different Brucella strains and different Brucella species, with a few kind of minor differences. And so that's all well and good, but the holy grail of genomics is not using just small parts of the genome, not just using a handful of loci from multi-locus sequence typing, or a handful of loci from VNTRs. We want to use the entire genome to look for differences. And so the advent of whole genome sequencing and particularly advances in Illumina sequencing have really made genomics accessible to lots of different people, and the ability to sequence hundreds of genomes and compare them against each other. But as we saw previously, kind of with that spider web of relationships, it does become pretty challenging, but we do, at our basics, kind of try to compare different chromosomes against each other. And I'm happy to kind of talk about exactly how we do that later, but basically we find we go in short regions and look at a particular short region of say this chromosome in this first one, and we try to find where that region or where that sequence is in other samples that are out there. And so it may be, this locus may be right here, and we usually do it in 100 base pair chunks. And so we look for 100 base pair chunk here, and then we find it, and we find it's actually here in this one, and then it's here in this one, and here in this one, here in this one, and here in this one. And so you get the basic idea where we can look at particular 100 base pair chunks and compare them against each other. They may be reshuffled across various parts of the chromosome of the different strains, but we can find what is orthologous, or what is the same region in all, in this case, all 6 of these different strains. And so doing that, so we did that on hundreds of Brucella genomes. And so lots of people like to show unreadable trees. And so this is my own unreadable tree. And so that's a bit of a phylogeneticist joke there. Lots of phylogenetics people like to show trees that you can't read. And so I will kind of break this down so we can see exactly what's happening. But we have so many genomes, at least in some ways of looking at it, you can't really see what's going on until you kind of focus on particular parts. But I'll break this down. And so we used SNPs, and so single nucleotide polymorphisms, or point mutations from across the genomes, comparing the same SNP locus across all of the different strains or genomes that we have in the analysis. And so this is the basic pattern that we see. And so this comes from a paper of ours from Rebecca Wadham in 2014, where we have 2 main groups. And so what we have is the classic Brucella up here. And so we'll go into detail about those classic Brucella. And then we had, at least at the time, several different Brucella that we didn't really know what to make of. And then we have the closest relatives. And so these are the Ochrobactrum. So 2 different Ochrobactrum species that are outgroups. And so we're just comparing the differences between Ochrobactrum and the Brucella. And we see that there's 2 major groups. There's what we now call the classic Brucella, all the ones we normally think of that are commonly found in animals. And then these more recently discovered Brucella that we really don't know what to make of them, and we're still kind of scratching our heads. And I'll show you kind of more recent examples in a second. And so what's interesting, and so go back, the classic Brucella are up here, are also known as the core Brucella. And so these are that classic or core Brucella here. And then these are a bunch of new genomes that we had. And so it turns out these ones out here that are outside of the core Brucella, the ones that we're finding more recently, share a lot of similarities to a lot more similarities to the soil bacteria, particularly the Ochrebacterium. And so some of them even have flagella, suggesting motility. And so the Ochrebacterium have those as well. And so they share a lot of characters in common with these soil bacteria. Yeah, we'll also get back to that later, but remind me if I don't do so later that that since they've diverged, these soil bacteria, from the soil bacteria here, and so presumably in the past, Brucella were soil bacteria as well, but over time they've lost many of the adaptations for soil. But some of these retain them, but don't get confused. They continue to evolve after they have diverged from the soil bacteria. So they are continuing to change. Just some of these kind of retained some of these ancestral characters. And so kind of more of this. And so this was even more recent. And so this was the rest of Brucella, including kind of those wacky groups that we saw previously. And so this was 10 isolates from bullfrogs that were imported from Africa into Germany. And so this was a series of 10 isolates of Brucella from bullfrogs from a single pond. And what we found after sequencing them was that in these 10 bullfrog isolates, I don't think all 10 are on this figure, but you get the basic idea. In 10 bullfrog isolates from a single pond, we had more genetic diversity than all the rest of Brucella combined, at least to that date. So we were finding more and more diversity as we were finding kind of the more of these wacky or atypical Brucella. In the grand scheme of things, they aren't atypical. They're just atypical to us because we're used to the classic Brucella that infect our livestock and infect us. So this is the most kind of recent phylogenetic tree that we've developed. And so this is again showing that classic or core Brucella up here. And so we'll go into detail for these, but it also shows that there's amazing diversity in this atypicals group. So these are coming from foxes. These are coming from rodents in Australia. These are coming from bullfrogs. This came from a breast implant in a human. We still don't know. This also came from a frog. And so basically we're finding all sorts of other Brucellas out there. And the more we look, the more we find. And almost inevitably, when we find a new Brucella, it's in this atypicals group and it's in a different host. And so a lot of these are in wildlife that are out there. And yeah, lots of unusual and unknown diversity. The more we look, the more we find. So let's focus in on the classical Brucella. In nearly all the work that we know about, at least in detailed genetic work and epidemiological work, and in fact, nearly all the Brucellas that you're going to be familiar with come from this classical or core group. And so to just put some pictures with the names, and so we have Brucella abortus up here, typically thought of as coming from cattle. It can cross into other hosts, and that may be a point of discussion later. We have melitensis, which is typically in goats, sometimes in sheep. We have ovis in sheep. We have a range of different marine mammal groups in both seals and whales and dolphins. And so we have this group of marine mammal ones here. We have ones that are suis, that are typically in pigs, and the one in dogs is kind of nested within this group. And then we have neotomi and microti. So this is the basic, some of the major groups of Brucella that we think about. There's some additional ones that we don't need to talk about, but this is basically the ones that are affecting our domesticated animals, and in a few cases, kind of some wildlife. And so I'll just point out, since we talked about time, something really interesting happened. And we don't know the exact date of this, using phylogenetics to date things, particularly when they're far back in the past and we have no fossil record, is imperfect. But what I will say is that this happened, thousands of years ago, likely tens, if not hundreds of thousands of years ago. This didn't happen very recently. So Brucella has really diverged in relatively long ago. So one question I'm often asked is, did Brucella diverge at the same time as its typical hosts did? And so did it diverge, tens of millions of years ago when its typical host diverged? And so the short answer is no. And so this is a phylogeny of a whole bunch of mammals, and the numbers here are in millions of years. And so one good example here is that we see in our kind of most recent phylogeny that that Brucella from seals and Brucella from whales and dolphins are relatively closely related and happened relatively recently, within the past 10,000 to 100,000 years. Whales and seals diverged tens of millions of years ago. And so this is just one example. An additional example is that the isolates coming from caribou and isolates coming from various rodents are really closely related to each other. In fact, some cases it's probably in the range of tens of thousands of years ago. And this represents at least tens, if not hundreds of millions of years of mammalian evolution. And so they clearly didn't diverge at the same time. The bacteria didn't diverge at the same time as the mammals did, at least not in the main hosts that they had. And just more and more examples of it not kind of matching up. So let's focus on one of the most common species that, or two of the most common species. These are ones that are common in Brazil, common kind of throughout Central and South America, common in fact throughout the world, as we're about to see. Brucella abortus primarily in cattle, and Brucella melitensis primarily in goats and sheep. And so this is a story about domestication, right? And so we've spread our livestock globally. And part of this is connected to using milk and dairy products as a primary source of protein. And so we realize that if we domesticate these animals and use their milk, we can utilize that milk and they'll continue to provide for us. And so as part of that, before we go into too much detail, the origin, this is a paper by Melinda Zader in the Proceedings of the National Academy of Sciences from 2008, that indicates that the domestication of much of the livestock that we think of, at least the mammalian livestock, happened in the Middle East. There are a couple of exceptions, and they're actually of relevance to Brazil that I'm happy to answer later, but many of the cattle that we think of, many of the common cattle breeds emerged from the Middle East. And we also see that sheep, pigs, and goats, all great hosts of Brucella, emerged, in the roughly 10,000 years ago. And so happened diverge, or at least domestication of these animals happened relatively recently. And so again, brucellosis and animal domestication centered in the Middle East. And interestingly, and so we're talking about this region here in the previous map here. Yeah, hold on. We're talking about this region right here as the center of domestication. And interestingly, this is exactly where brucellosis is still centered. I don't know if that's just a coincidence or not, but still, so animal domestication and brucellosis are both centered in the same place. And so some of it's coincidence and some is not, and we'll see kind of some of those details in a second. And so we have taken from those centers of domestication, we have moved cattle and we have moved goats and sheep globally. And so from that single location, or potentially two locations, we have spread these livestock to all 6 of the main continents. And so let's just look at goats. And so this is a distribution of goats. Sorry for the quality of this image, but we see that we've spread goats, globally to lots of different places, and cattle even more so, that we'll see in a second. But we've really spread goats throughout the world. They're concentrated in particular areas, but they're on all of the major 6 continents. And so how did it get there? And so hopefully it didn't get there on the backs of people, but I guess I wouldn't rule that out. I don't think it got there by little boats, but who knows about that. But the fact is that as we've spread our goats globally, we've brought along Brucella. And so Brucella, in this case, Brucella melitensis is in goats. And we go back and use VNTRs, or the VNTR system, or VNTR analysis called MULVA, to look at the relationship of different Brucella isolates. So this is work from a collaborator of mine, Giuliano Garofalo, in Italy. And what we see here is 3 major groupings. And so lots of other people have found these same groupings. We see a grouping here that corresponds with the East Mediterranean. And it's a bit confusing. It actually starts in the East Mediterranean and goes all the way to East Asia, all the way to China and Japan. But this is kind of, at least the early isolates that we found were all in the East Mediterranean. Then we later found out that this is really kind of goes all the way throughout Asia. But the basic idea is that we have this one group here in blue. We have another group here, the Americas or Iberia group. It's all here in red. And then we have this third group that's usually in black, although in this study, we added additional isolates from Italy. This is what we call a West Mediterranean group. So in general, Brucella melitensis falls into 3 distinct groups using VNTRs, and we can see the relationships of the different groups. And so just to make this a little more interesting and give some examples, in this case, this was a sample from a patient in Italy. And it turns out, and so it's really weird, this was a patient from Italy, but infected with a strain that falls in the East Mediterranean group. So we're trying to figure out, how did this happen? And so if you actually go and do the epidemiology, it was determined that this was a patient, I believe he traveled back to Syria I believe it was Syria. He traveled back to the Middle East and was infected when he was back at home, and then brought it back to Italy and was diagnosed in Italy. So clearly this is not an Italian strain. This comes from the East Mediterranean group, and we can use VNTRs to identify that. So both big scales and happening over thousands of years, and really fine scales happening within a year. Another example here, these were isolates from goats in Italy. And so this was a head scratcher as well. They all fall in the Americas or Iberia group. And so Iberia, well, you all probably know this better than I do, is both Spain and Portugal. And I think you can probably imagine how Spain and Portugal may be directly connected to the Americas. And if this were an open class, I would ask you all to answer that. And so, but interestingly here, so these were strains from Italy, from infected goats in Italy, but they fell in this Americas or Iberia group. So it turns out these actually were goats that were infected. They were imported into Italy. And I believe they came from, I need to remember the details, but they came off the coast of Spain. And so these were imported goats, and we could exactly tell that they came from, either the New World or Spain or Portugal, just based on the genotypes that they had. So a small aside, thank you for permitting me the license to do that. And so that was using VNTRs. And so we get those same patterns when we use whole genomes. So again, VNTRs are just 16 loci or 16 small parts of the genome. Now we're using the entire genome, all 3.3 million bases. And so we get those same groups. So we get this Americas group. And so this is a commonly known strain called 16M. This is America's group here in red. We get this same East Mediterranean group here in blue. And this again kind of goes from Eastern Mediterranean all the way throughout most of Asia. And you'll see kind of some country codes. There are a few exceptions in here because of likely movement, say, for example, Mexican strains here, but in general, we have a pretty strong pattern of geography with the particular lineage that you have. And then finally, we have this West Mediterranean group, also known by one of the type strains called ether. And this is all centered around Italy with kind of a couple of exceptions. And so this is part of research done by a group out of the UK, lead author with the last name of K. And they're actually able to generate partial genomes from skeletons that came from Sardinia. And so they were able to sequence ancient DNA and then place it into this phylogeny. And they're able to identify that that the strain or the genome from the 1300s falls into this group, this West Mediterranean group that's still in that same location today. It also gives us, and so we have evidence that this strain or this lineage of Brucella melitensis has been there for nearly 1,000 years, and that we also can estimate roughly how much it's changed over time. And the short version is that it hasn't changed much at all. And so Brucella is relatively slowly evolving and has been in this region for a relatively long period of time. And just another example, we've done additional work in camels, trying to identify kind of the source of different groups. So it turns out actually that there's a fourth group that we didn't recognize at the time, but it may be obvious now. we see kind of major groups here in green and blue, but we see two groups here. So we see two clusters. There's one here of this group in the top part, and there's an additional group here. And so the more we looked and the more we recognized that nearly all these isolates came from Africa, with a couple of exceptions, so Great Britain, but actually they were only identified in Great Britain, not coming from Great Britain. But the basic idea is that these were African isolates, and we determined that this was actually a new and unrecognized group that we now call the Africa lineage. And so that is Brucella melitensis, and kind of a bit of a background how we can use different genetic markers to look at the evolution of one of these groups. And so perhaps even more relevant to Brazil and much of the world is that we've spread cattle even more geographically extended than we have goats. And we've spread them again to 6 continents, but throughout, much of the world has been created into pasture, or at least growing crops to feed cattle. And so we've spread them globally. And at the same time, Brucella abortus spread with them. So Brucella has spread to all 6 continents, likely as we transported cattle. And so this is all about the global movement of Brucella abortus and how we can use genetics to kind of track that. And so what we see here is, in general, again, kind of a phylogeneticist joke where we put all the strains on a single tree, and we can't see much, but I'll go into kind of some of the details here. And so we see here that we have these are the common Brucella strains that we think about. Indeed, most of the strains of the world fall into this group, and they're all very short branches, indicating that they are closely related to each other and that this happened very recently. And so interestingly, these come from a global collection, strongly suggesting that a single group of infected cattle were spread globally and very recently. And then we have these other strains out here that are a lot more diverse, and we know that just based on the branch lengths here. And so these are the biovars. We can talk about that later as well, but this is just a biochemical approach where we can do various groupings. It does an okay job in Brucella abortus. It does a terrible job in Brucella melitensis. But the basic idea is that we have these groups of biovars, and they're diverse, particularly they're very diverse compared to this kind of common group. And many of them, although not all of them, many of them come from Africa. And so I'll show you that, show you more in a second. And so these are just the 4 major groups that we've identified. They correspond to different groups that we saw in the previous tree, but this is just a more readily interpreted phylogenetic tree that you may have seen previously, or similar ones previously. So these 2 groups that we saw previously, these all or nearly all come from Africa. They're really diverse, and they're really diverse based on the long branch lengths, and they're very different than at least the other 2 major groups that we know about. And so if we look at geography, nearly all the isolates that come from A and B come from Africa. This group C, nearly all of them come from Asia and the Middle East. And then nearly all isolates that we have here in this group that we call Group D come from Western Europe, North America, and South America. And so in general, we have strong geographical differentiation, and we can identify a likely kind of source of different Brucella isolates depending on where they came from in the world. And so this is just for the folks who are really into kind of Brucella phylogenetics. But again, this is those groups A and B, and they're really they're really genetically very different than much of the rest of the world. This is group C, that's from the Middle East and Asia. Again, kind of showing it in comparison to the other groups A, B, and D. And then finally, this is group D. And it's impossible to see because there's so many strains in this phylogeny. But the other main point I kind of want to show is that they're very, very similar to each other. And so most of these strains are only differ from each other by a handful of SNPs. And so, and the whole, in the entirety of the phylogeny, there's only a few hundred SNPs that differentiate the hundreds, if not thousands of strains, indicating that it happened very recently, and that so the expansion of this particular group, happened within the past likely several hundred years. And so I don't know how much time we have left. I will, Elaine, perhaps you can suggest. I do have a kind of small case study from Yellowstone if you all are interested.
We are planning an interval
okay.
From 25 minutes from now.
Okay, great. All right, so I will show we're talking about in kind of the previous slides, or at least a lot of them, kind of the global spread. And so in this case, we're doing a fine scale. So global spread over hundreds, if not thousands of years. And in this case, we're using a much more recent kind of case study. And so this is a famous case in the United States of Yellowstone. So Yellowstone was our first national park, and it's a really amazing place in Wyoming, Montana, and Idaho. And so the greater yellow ecosystem has some of our biggest grazers, but at the same time, there's livestock that are grazing in these same lands. And so at least in the past, the bison have been blamed for giving brucellosis to the local cattle and causing abortions of cattle. And so bison were in fact shot on as they were trying to leave Yellowstone National Park. And so maybe not by this gun, but you get the idea that, there was a big confrontation between ranchers and people who wanted to conserve bison and other wild animals. But the ranchers were concerned that the bison were giving Brucella to their cattle. But also note, at least in this picture, at the same time, we have elk. And so this is known as red deer in Europe, but these are our second biggest deer species. And so elk are absolutely enormous, and they're grazing or browsing in these same areas as the bison and the cattle. And so lots of people like to blame the bison, but at the same time, we know that elk are also infected in these ecosystems and may be responsible, although they're not being shot at the entry of the park. And so this is a study led by a colleague of mine named Pauline Kamath, who's now at the University of Maine. And so we analyzed 245 genomes that came across a range of dates of nearly 50 years. And all these strains came from the greater Yellowstone ecosystem. So here's Yellowstone National Park here, but it's part of a larger ecosystem that's all interconnected. And we can see we got isolates in blue that came from elk. We see isolates in red and orange that came from wild or farmed bison. And then we see isolates that came from cattle. And so we're really trying to understand, are the bison giving Brucella to the cattle, or are the elk giving it to the cattle, or how is this exactly working? So again, 245 genomes looking at the relationship of different isolates across this entire kind of region. Again, we have 3.3 million base genome, but there are only, it's actually quite a bit for this type of study, 1,400 SNPs. So we had 1,400 different loci that we can use to look at the relationship of different isolates in this region and see who is infecting whom. And so I won't get into all the various details here. We can use a molecular clock to understand the timing of it. So again, mutations accumulate roughly at a constant rate with, a bit of hand waving in there. And so we can see here, we've dated when various things occurred. And so the likely introduction of Brucella into this ecosystem in roughly the 1960s, and then different lineages that have evolved. Again, kind of similar. This one, may be thought of as the Delta variant for SARS-CoV-2, but you get the basic idea where different lineages evolve over time. We can see here that at least, roughly since 1960, at least 5 different lineages were introduced to the Yellowstone ecosystem, and we see those today in various parts. So this was the ancestor roughly 50 years ago, which is interesting because it disagrees with the known introduction of brucellosis roughly in the early 1900s. And so the suggestion is that what was initially introduced has died out and is no longer present, and that the ones that we see today were introduced more recently. But pretty wild that kind of these widespread lineages that we have were introduced very recently, and we can use SNPs to look at the relationship of the different ones. So we have 3 different host species here that we see in the different slices of the pie. Black are coming from elk. So we see here that this is largely an elk lineage, but it's also found in white cattle and gray in bison. In this case, we have this one's largely in bison, but it's also in elk and cattle. This one is kind of a more even mix, and this one is a more even mix as well. So different places have different kind of proportions of who's infected with what particular lineage. So we have both widespread lineages and we have localized lineages. For example, we have a really localized, this one in purple here, Yellowstone and Paradise Valley is really localized here. We have kind of one exception here, but really localized to this particular area. And the feed grounds here, so they feed the elk in winter here, are largely localized here. And we see these other feed grounds, one here in green, and so corresponding to these 2 groups here. So localized. And then we see 2 widespread ones. And so the gray ones and the blue ones are widespread throughout nearly the entire Yellowstone ecosystem. So what we have here is that we have cattle, elk, and bison all giving Brucella to each other. And it's not solely that the bison are responsible for infecting the cattle. And so, yeah, that's just a short kind of snippet of how we can use single nucleotide polymorphisms to look at fine scale within the past 50 years of the relationships of different isolates and understand kind of who is infecting whom. We did a series of kind of phylogenetic reconstructions to understand and look at transmission. And it's based on a probabilistic framework. And in nearly all cases of infected cattle, it was most likely that the elk were infecting the cattle rather than any of the other animals infecting cattle. So it seems like, at least based on the data that I showed previously, that it's elk infecting cattle and not vice versa. And so a quick overall summary, and then I'll take questions. I imagine I skipped various parts that you want to hear more about. But Brucella evolved from an ancestor in the soil. All of its relatives are in the soil, and it's lost many of the adaptations. I didn't go into this in a lot of detail, but it's lost many of the adaptations that are associated with living in the soil, and has developed a bunch of different adaptations that allow it to survive as an intracellular bacteria within vertebrate hosts. So it's both lost things that allow it to compete in the soil, and it's evolved or adapted to do well inside of vertebrate hosts and to take advantage of the resources in the host. It's diversified and expanded globally. And so as a genus, it's done that, and several of the species have done that as well. Some of them are certainly preceded domestication. And so, for example, the ones in marine mammals, marine mammals are not domesticated, and yet Brucella was able to spread globally. Exactly when and how that happened, we still don't know, but it happened tens, if not hundreds of thousands of years ago. And so Brucella lineages, I'll just remind you that they continue to evolve after they diverge. So if a point I'd really like to make, and others have made it as well, is that our ancestors are no longer amongst us, at least in a, not in a metaphysical sense, but in a real sense, right? And so our ancestors are no longer around, only their descendants are. And so even after things have diverged, they continue to evolve and accumulate changes. And so that's the challenge of phylogenetics. We're only seeing what's around now. We're not able to see into the past, with a few exceptions, such as that getting a genome from a bone from a skeleton from the 1300s. That is an exception, right? And so, but we rarely have kind of that ability to do that. You've seen two examples of using different molecular markers for different timescales. In general, VNTRs evolve more rapidly, and they're really great for kind of more recent divergence. But sometimes SNPs can do that as well. And so we saw kind of divergence over the last 50 years in using SNPs to look at the relationship of isolates within the Yellowstone region. But sometimes SNPs don't evolve quickly enough. And so we need VNTRs to kind of look at that. So a lot of the kind of recent epidemiological studies, and you saw some examples from Italy in both the goats infected in Spain and the patient in Italy infected in the Middle East. We can use VNTRs to identify the likely source of infection. Also say that we found unexpected diversity, particularly in those groups of Brucella abortus within Africa. And so there's lots of diversity in Africa, likely underappreciated, and it strongly suggests that our sampling is not that good. We've been really biased in how we've sampled, and we've really sampled our livestock really well. And so part of that is that our cattle and goats are getting infected and we want to know what's happening there. So we collect isolates. So there's a bias there, but it's also in terms of abundance. We have billions of goats and cattle. And so we're interested in their welfare. And so we're looking there. But whenever we look elsewhere, we're finding Brucella. And this includes a whole range of other different vertebrate hosts. And interestingly, Brucella abortus as a species is younger than previously estimated, and it occurred well after cattle domestication. And with that, I will take questions, and thank you for your attention.
Thank you very much, Jeff. Was a really nice talk. Thank you. We have some questions here in the chat. I think we have 4 questions, 5 now. The first one is from Janira. She's asking if the Brucella genus shares a common ancestral with soil bacteria. So how can you explain the marine Brucella strains? Did they emerge after? So the strains come from the historical hosts?
I wish I knew the answer. We don't. If we go back, at least in our memory, to think about, maybe I can go back. One of the big challenges is exactly what happened in this rapid radiation of Brucella, and we still don't know. And part of the difficulty there is that it happened so long ago that we don't really know what happened. And so what seemed here is that it happened almost instantaneously. It diverged into all these different groups. And what's difficult is that it happened long enough ago that that signal has started to erode, and that we don't get a lot of differentiation. So we don't really know how that happened. And did that happen over 100 years, or did that happen over 10,000 years or not? And so we can make some rough guesses about how long ago it happened. It probably happened a few 100,000 years ago, but certainly a lot more recently than the animals themselves had diverged. So it's in the hundreds of thousands of years, not in the millions of years. But what exactly happened, and why did it go into marine mammals, and why did it grow into terrestrial mammals, and how did that happen? Your guess is as good as mine, and I would love for someone to come up with better reasons than I have.
Thank you. Also, Saulo is asking, can you say that the Brucella basically evolves and mutates for host survival rather than competition survival with other bacteria, for example?
Yeah, so I think, I'd say yes in general, although we need to be careful about how we use that terminology, because it's mutating, right? And some of those mutations give it an advantage, but it's not intentionally mutating. And I know the difference is subtle, right? And so mutations are occurring, and some of those mutations give it an advantage in a particular host. And I think we have clear examples of that happening. And so it's clearly better and has particular adaptations for host survival inside host cells. And we have some evidence that different Brucella have specific advantages to the hosts in which they're normally associated. And so that's best example is Brucella ovis. It has particular kind of mutations that allow it to do well within sheep that don't exist in the other Brucella. But this is again another major point of research that we don't really know why is Brucella host adapted, and how host adapted is it? Why did we find Brucella abortus typically in cattle? Is it because it has particular adaptations that make it better in cattle? We have some suggestions that that's true, but other times we know that we can infect, most of these mammals on this list, if we give them a shot, we can infect pigs with Brucella abortus. We can infect cattle with Brucella suis. And so it may be just the likelihood of transmission, or it may be both. And this is the kind of second major area that I wish I knew the answer to. And so I love the question, that it's a big open question about host adaptation and why the different Brucellas appear to have host specificity, although it's not complete. And so thank you for that.
Thank you. And Karine, she's asking, she could ask you personally, but she's asking here, is the identification of atypical Brucella only in the recent years related to their lower ability to infect domestic animals? What would be possible strategies to look for more of these atypical isolates?
Oh, I love this question. So I would say it's not a lower ability. I would just say that, yeah, it's really, we're finding them because we're looking. And we found the classic ones because we've been looking, because they're the ones that are around us, right? We're looking at our dogs because our dogs get infected, and we want to know why, our dog is having an abortion. We want to know why our cattle are sick. We want to know why, drinking cows or goat's milk has suddenly made us sick, right? And so part of it is that we're looking for it, and we look for it in the animals and the infections that we have. And so it does seem like there is that some of these strains are less pathogenic. And so we get and aren't able to infect a broad range of hosts. I would also say that we don't have great evidence of pathogenicity, although it's there's a suggestion that they're less pathogenic, and we're also finding them just because we're looking for them, and we haven't looked for them in the past. And so, yeah, I love that question. And so, what are possible strategies to look? Basically, anytime you can find a wild animal, I would look for it. And so it turns out, yeah. So I had this idea. I used to be in New Hampshire, this idea about finding roadkill and doing necropsies on all roadkill to try to find these atypical strains. And I think it's going to be everywhere. Yes. So we not talked about here, I do lots of work on bats, and a new bat, a new species has been found from bats. And interestingly, it's nearly identical. So this was found in the Republic of Georgia, in Asia, Western Asia. And it turns out it's nearly identical to rodent strains isolated from that exact same spot in the 1960s. And so who knows what's going on there? It's probably, but a bat isolate nearly identical to a rodent isolate from the same location. So it's just a case of looking and knowing which tissues to look in. And so there's particular, there's tropisms for particular tissues. And so it seems like lymph nodes and liver are good places to look, but it depends on the Brucella sometimes. So yeah, I love that question. And again, I wish I had a better answer. These are all, yeah, you're getting to the root of all my insecurities. I don't know the answers to a lot of these, and but it makes it exciting too.
It's a nice discussing.
Yes.
Kathy is asking, could you think of an association between climate change and the appearance of Brucella atypical variants?
I don't think so. I think they've always been there. We're just finding them. Again, this bias, right? There's a bias in that, you know, we have all these Brucella livestock isolates because we're looking. We look in our domestic animals, and I think they've always been there. Well, they've been there for thousands of years, if not hundreds of thousands of years. We just haven't found them.
Raquel, she's asking, this Brucella evolution from soil to pathogenic should warn us about potential of the same occur with similar genus, with I think similar species from the genus.
Yeah, I'm not too worried. I mean, it takes a long time for this to happen, right? And so I'd be more worried about the ones that it has happened in. Yeah, it takes a long time for evolution to happen. And so it's unlikely that Brucella evolves very slowly. And so it's unlikely to happen within any recent times.
And we got a last question from our colleague, Professor Geraldo. He's asking, first, he congratulates you about the lecture. And does the present knowledge allow us to think about a universal vaccine to control brucellosis among animals of production animals?
Yeah, that's a difficult question. So I will say that there's $1 million prize out there for the best. I think it's in goats, and it's not even a universal one. It's just ones in goats. Vaccine out there, and no one has won the prize yet. And
but so the requirements are too high for the
yes.
They are asking for.
It is. The short answer is vaccines within Brucella vaccines are complicated, and a universal one, certainly there's several groups out of Argentina that have promising universal or nearly universal vaccines. It's unlikely because of the phenotypic differences that we see in some strains. For example, there's rough versus smooth strains. The vaccines only work on one or the other, and so I think it would be more likely that certain groups would be vaccinated or vaccinatable. So say Brucella abortus and melitensis. And so I'd say the genomics only gets us so far, and it turns out we can't, at least genomically, we haven't been able to find those specific targets. We know how they're related, but their connection between genetic variation and immune function is too distant, at least at this point, to really have effective vaccines being guided by genomics.
In this question of Geraldo, I have another question. We, Karine and I, and Andre, we have studied about vaccination and mutants that they have made, deleting one gene to make HUF strains, genes related to the OPS exportation and synthesis. And some of the papers, they found that depends on the parental strain which you use to make the mutants, you have different efficacy of the vaccine tested. It seems that the genetic background of the strain counts on how this mutant, this vaccine candidate can stimulate the immune system. I was very intrigued by this found of this paper.
Yeah, I would say this is at the edge of my knowledge, but I will say that there's been some really interesting vaccine-related studies. And so what I think one should do is, and a few people have done this, perhaps not as exhaustively as needed, is to really look at what is the full range of genetic differences of vaccine strains. And so there's a few common vaccine strains out there, RB51, there's S19, there's Rev1. And so it depends on who you're vaccinating. But some really interesting studies in that in that passage of the vaccine while you're creating it has created new variation. It's subtle, right? And there's not hundreds of SNPs, but those handful of SNPs can have effects on genotype. Well, they have genetic differences, and occasionally they have phenotypic differences, and they're not recognized until far later when you kind of see this range of different efficacy. So I'm not surprised by that at all, that we're having genetic differences. Yeah, at the time, right? And then you're like, oh, it's making sense.
Yeah.
They're small but important differences. Most of the differences don't make any difference, but occasionally they do.
Yeah, I'm seeing that in Brucella, studying the genomes. I can say that it's really hard to find what is important in Brucella, much more than in enterobacteria and other bacteria we work with, because it's the details make a lot of difference in some time, but it's difficult to find which detail we should look at.
Yeah, I totally agree. And it makes it's frustrating because compared to most other bacteria, Brucella is really simple. Like, not a lot is happening. We should be able to figure this out. There's only a few hundred SNPs, and they're so similar to each other, and we still can't figure it out.
But it seems that
infuriating.
It makes more difficult than simple.
Yeah.
I agree.
Yes.
Thank you, Bev. Thank you so much for the talking. I really enjoy, and I'm pretty sure everyone enjoy.
Yes, thanks everyone for your attention and wonderful questions, and I really appreciate being invited.
Thank you, and we are coming back, people, 4:30.
Okay, thank you.
Okay, bye. Nice to see you there.
All right, bye-bye.